Right, ecological change and ecological science. So this all starts with our science of ecology, which is, we could say it's maybe 100 years old, give or take, a little bit more than that. Most of the pillars of this science and our concepts and most of our models were built at a time when this is nature. Nature was pristine, it was wild, it didn't, we weren't part of it now, or maybe we were before, we weren't very clear on that. So ecology's goal, where as an ecologist, the science I practiced came from a place of being dedicated to understanding how nature works. What are the processes that explain the patterns or cause the patterns that we see? So as an ecologist, these are the tools I use. And they're the tools we're all using if we're doing ecology. However, most of us see the world this way. As people, as citizens at this time and place in our planet, we have this idea that nature is changing fast. And we actually have an idea about why it's changing. It's changing because one species, us, has become a super predator, something that our theory didn't have a place for until recently. And that same species has redistributed resources globally. So even in the context of our classic theory, we know that something very different is happening on the planet right now. And most of us, when we give our talks, we write our introductions, we write grant proposals, we are using a science that was built to understand, but we claim now we're using it to forecast, predict, and project the future. Now, philosophically and technically, these two are very different kinds of aims. You use different tools and different kinds of inference to understand than you do to project and predict forward. And ecology is getting better fast at projecting and predicting, but I think we still have some confusion about how well the science that we were trained in is suited to do what we want it to do. So one thing that I think we've done without maybe fully realizing it is we've actually redefined the science that we do. Our science of ecology, and if you read textbooks, you'll read that it's about interactions between organisms and their environment. It's about explaining patterns in the abundance and distribution of species. But I think what we're really doing now is practicing a science that's dedicated to understanding how living systems grow, change, and persist. The reason we want to understand that scientifically is because I think, you could argue with me on this, but one major way we're going to say something about the future is by understanding the dynamics of the systems that exist now and have existed in the past. So that's growth, change, and persistence. So if this is what we're actually doing, how are we doing it? How well are we doing it? And I get really frustrated sometimes, and really excited also, but frustrated when I go to a meeting like ESA, Ecological Society of America, which will have 4,000 ecologists, and almost everybody there is an ecologist. And yet, if I encounter someone in the hallway who I don't know, it takes quite a long time to establish a common starting point in our science for when we could then share kind of what we're doing. We have different assumptions, we have different goals, we have different backgrounds. A lot of this has to do with, under this umbrella of a science that we call ecology, we actually have many different disciplines that were happily distinct and different in recent decades as they were grown and developed. And now we're doing much better at building bridges, but we have ideas, we have theories, we have assumptions that belong in one or the other of these domains, natural history or systems ecology or community ecology, that don't all actually align or match up neatly with what we might assume or do in the others of these disciplines. So I think that the consequences of this fragmentation of our science's history have led to great opportunities now for ways we can do science to understand our changing world. So this is where my motivation and my real interest in unifying our science comes from. It comes from building bridges and if an idea or an assumption or a finding from one discipline doesn't agree with another one, we should figure out why, because there's an opportunity there to build better knowledge. Okay, so that is one motivation for a basic, a focus on understanding how nature works at very basic levels and refining our understanding. Another motivation I have comes from my experience working with how science is used at global levels and local levels. I have been a contributing author both to the IPCC and the IP Best report, so I have seen how the science that we might do makes it up through the various filters and gets into these reports. We know that, and many of us are probably hoping that what we do would influence something at this level, right? So the way that that happens, in my experience, is that first of all, you have assessments of change. So you pull together the state-of-the-art scientific information about change, whether it's climate change, ecological responses to climate change, or whether it's biodiversity change. And we hope that that influences public policy and understanding. But to get there, we have to use scientific effort, scientific work that both detected change and attributed that change to some driver. Human activities usually is what we want to know about. So those are right there, non-trivial major efforts, and there are knowledge gaps even there that are pretty serious right now. But even before we detect change or try to attribute it, we have to do analysis, synthesis, and inference. We have to do basic scientific methods. If we're going to do that, we've got to have some data. So we have to have observed nature and collected data. Before that, we have to have expectations for what we're going to observe. If we go out in nature or we design a sensing system or an observation system to observe nature, we're doing that because we have some expectation of what we want to know about. We want to know about biodiversity. We want to know about phytoplankton abundance. We want to know about primary production. So we design the system that we think would give us that information. So data are not the beginning, and data are not the end, and they're not truth or untruth. They're the thing that we use because we expect something and we want to go learn about it. So these expectations are really important, and those come from scientific understanding. They come from elsewhere too, social context. They come from our personal values, but that's another thing to talk about. So for today, we'll talk about the expectations for what we observe, how we enter into this process, comes from our scientific understanding. And I have seen, I'll give you two quick examples where I have seen that I believe it's the scientific understanding and expectations part that actually have been a limiting step to the whole rest of this chain of events. So another way to say that is if you're limited early on in this chain of science methods or scientific actions, you can't recover later on. No amount of attribution statistics or analysis that can make up for insufficient data or faulty expectations. You can't rush to the end and hope to fix it by doing fancy stats. Yes, and of course, this is simplified. All of these things affect and feedback on each other, for sure. So I've already kind of suggested that the ecology that we've had for the last 50 years has gotten us very far, but hasn't gotten us quite fully prepared to think about change. And so then I'm just this is one of the reasons where I came to this idea that if our science is about change, then it's got to be integrative, and that's where we should be drawing our expectations from for how we look at global change in the past and the future. So one of these examples I said I would tell you about came from working on this paper. This was a working group that met for over several years at NCEAS, and the motivation that previous IPCC reports up to the fourth one really didn't include marine time series. They had something like 30,000 terrestrial time series to demonstrate climate impacts and literally like 28 marine biological time series. And that was because there just weren't marine ecologists, biologists, authors on that panel that were doing this kind of work. So this working group did the work for them. We spent 3 years doing this, and this is basically one of the flagship figures that ended up then in the fifth assessment report of the IPCC. And here's the best we could do. Okay, so what this is a map from everywhere we found in the literature, time series of marine biological attributes. So abundance, diversity, those are basically the two big things. Phenology. And we followed the IPCC's criteria for inclusion as evidence. So time series have to be 19 years long. There's other criteria. So we followed their rules. And we came up with these this is where the studies came from. But then the next challenge was attribution. IPCC wants to know not only what has changed, but was climate change possibly the cause? Well, how do you know that, right? The best we could do after weeks of arguing and discussion and reading was we had to go with the author's expectations. So in every one of these papers, which may have been written any time in the last century that has any kind of time series of abundance, and they had to have at least one climate driver. So maybe they had a temperature time series, was pretty common. We had to search those papers for the author's stated expectation for what climate change should have done, and then their assessment about whether that change was consistent with climate change or not. Okay? And most papers did have some kind of explanation, expectation fully half of them, and I know that because I'll tell you about what we did next, were post hoc discussion points like temperature changed, phytoplankton changed, it must have been climate change. So we counted that as an expectation, but that is scientifically very weak. But that's the best we've got. This is how we do it. We're trusting the scientists' own expectations for climate change. Some studies, to be fair, of course, do a first-rate job, and they do proper attribution, and they reject alternate hypotheses, and they do it great. Many do not. So what we did then, because this bothered us so much, we actually have no idea how good this is. We then went back and we created our best effort at an objective score to consider how did authors state their expectations, where did they come from, was their data suited to testing those, et cetera, in different statistical methods. So we did all that, and we were able to then come to this conclusion that most studies in the climate literature aren't using strong inference for attributing to climate change. Doesn't mean these biological effects aren't due to climate change. It just means that if you don't believe me and I'm going to try and convince you based on this evidence, I'm probably not going to convince you. All of us on this working group were lifelong marine ecologists, and all of us would agree with what I just said. We were really surprised that the actual robust evidence just wasn't there for time series. And I should just say, many of the authors of these papers, even when they made their post hoc explanations, would say that they clearly didn't have good basic science to link climate phytoplankton abundance, for example, to temperature. They just assumed that one causes change in the other. The other one recently that I've been involved with a lot and came to basically the same conclusion here was this. It has to do with changing biodiversity. So we have lots of evidence from how we understand nature to work that biodiversity is declining. Science and Nature papers with titles like this one, Defaunation in the Anthropocene. So we know from how we know nature works that we think we have a pretty serious biodiversity crisis. And yet, papers can be published in PNAS and in Science claiming no net change in local biodiversity. And they're using data, and they're finding, if you look at how at the local scale biodiversity has changed, you see lots of places where it goes up and lots of places where it goes down, but on average, there's no net change. So that may be perplexing, but what really got me is actually that shouldn't be perplexing. We've got lots of good ecological theory that says species richness is pretty stable at the local scale. So we actually should have expected that. These people, who I have worked with since then, these people literally wrote the books on biodiversity, and in their science paper, they have this statement. Whoops. They say, surprisingly, we did not detect consistent negative trend in species richness. They should not have been surprised. They were surprised because there was a social expectation, sort of a cultural expectation they were drawing on, and let themselves, as scientists, claim to be surprised. So, and this is okay, this happens all the time, I think, in our ecology. So this is another one which recently kicked me all the way back to getting our basic science right and using that when we're trying to understand global change. So that's a long intro, but I do it because I'm going to tell you about really basic stuff today for the rest of the talk. But I actually think for me, it's my best contribution to trying to understand how our world is changing and what we might be able to do about it. Another thing that I value as I think about this is, of course, if the link between basic understanding of nature and public policy not only goes through lots of these steps, but nature is complex. We have to understand a lot about how it could work, and we're not going to understand all the details. So another thing that I value is return on investment. So if we're trying to achieve a goal with our scientific knowledge, we're putting in effort, our own time, our own effort, our own prioritization. Am I going to work on this project or with these people or those people? I want to get as much bang for my knowledge buck as I can, right? And so this, you'll see that this pushes me into what I call information efficient approaches or theories. How little do I need to know to be able to know something about how the system is going to change? Okay, so that's the setup. I'll very briefly sort of give a slightly more specific proposal for why what I think this unified ecology might look like. Of course, I don't have the answer, because that's a big job. And then I'll tell you about some of the research going on in our lab, specifically in the context of temperature. And then that's what we'll do. Okay, so our ecological, I mentioned that ecology, in my mind, is now the science of living systems. So I thought if I really wanted to understand that better, I had to actually think we have to focus on growth, change, and persistence, but we actually also have to focus on living systems. So what is a living system if it's sort of the meat of our ecological science? And I had a sabbatical last year, so I actually did get to spend a bit of time going down this road, and it was super fun. So you should do it if you haven't done it recently. I broke floors where I thought I would dig and think I'd get an answer, and then I never got there. So here's what happened. There's two scientific views on what life is. Two of them, not just one. There's probably more, but there's two that I think count as major pillars. One of them is this idea that living systems, or living, is metabolism. It's the harvesting of energy and processing of materials along physical energy groups. Most of these would exist anyway without life. It's just that life has found a way to make them go faster and get some, use that energy for other purposes. So this view of life, I would argue, does underlie a lot of our major ecological theories and practice. Like ecosystem science is solidly in a metabolic view of life, right? So are bioenergetic models, the idea that it takes energy to power, and let's look at what happens. So these are all many aspects of food web ecology fit in sort of an energy materials view of life. But some of you are definitely thinking, well, and I've had many people say to me, well, I just don't care about carbon. What if I don't care about carbon? I want to know about biodiversity. Well, then you probably subscribe to this other view of life. That's that living is information processing. This turns out is not just biological. The physicists, the astrobiologists, everyone out there who's scientifically trying to figure out what life is, the people who wonder if computers are alive or can be alive or will be alive, they're using that definition of life. Information processing first. And information processing at some point, of course, requires energy and materials. But when we do that, we're bringing in the energy materials as a secondary component, a power source for what life really is, information processing. Just like when we do this, we say, well, of course, we need genes, we need organisms, we need physiological systems to metabolize the energy. But in that case, we're often bringing in the information processing as a secondary way of explaining how we get this metabolism. So what I was surprised to learn and a little bit disappointed, because I kind of wanted someone else to know the answer, is that these two things aren't fully reconciled. We don't know whether life is one or the other or equally both. And by we, I think I mean anybody. So we can rest assured that our problems in ecology, this giant gap that we have in the way we view nature, whether we view it by energy materials or by biodiversity, that's a real knowledge gap that goes down to the very roots of what life is. And I've been really frustrated with myself for the last few years because our lab works on biodiversity and we work on metabolic scaling, and it's grown into these two large, successful research programs that are frankly more than I can handle. And I've been trying to put them together, and I can't, but it's not my fault. They really don't go together yet very well. Okay, so I'm going to explain a bit more. I want to take the time to explain a bit more this information processing idea, because we all work on it in some way, and we all work on the energy material processing in some way, and I'm really trying to understand if this is a way we can think about ecology differently. So I'm hoping that if I give you a little bit more illustration of that, because we do get to see each other as often as we want around here, I would love to talk to you more about where this could go, and if you think it's helpful or if you have feedback for me on this way of thinking. Okay, so in the metabolism is energy and metabolism of energy and materials powers life viewpoint. Let's use this freshwater system. And though I'm a marine ecologist, this work is way easier to do in freshwater, because when you warm up freshwater, you don't have to deal with salinity and acidification and all this other kind of stuff. So now my cartoons and experiments are freshwater. So if we think of metabolism, and really what I mean by metabolism is these very fundamental, highly conserved metabolic processes. So carbon fixation, oxygen production, if we're going to use sort of macroscopic biological systems. So that happens in our mitochondria and chloroplasts. These things flux oxygen and carbon, and they do so using pretty much the same machinery to do those conversions. And on top of that is all the other metabolic processes that constitute the life and work of these organisms. But we're just going to focus for now on oxygen and carbon fluxes mediated by photosynthesis and aerobic respiration. So these are happening, we know, of course, at the organelle level. when we measure these fluxes, the biological parts of them at the ecosystem level, we're just measuring the sums of what's going on in these organelles. So we're pretty comfortable with that. We can also track carbon and look at food web dynamics and other ecological processes by tracking carbon and isotopes and things like that. But moving on up, we can also look at fluxes of energy and materials in terms of metabolism at the local scale, at larger scales, and even at the global scale. And when we do that, we understand that these global fluxes are mediated by individuals. And we can kind of scale up and down in the way we understand this right now in our science. Okay, but what about living as information processing? How does that work as sort of a cross-scale pillar for our ecological theory? So we do have information at all scales in nature, right? So take a minute and think about where you expect me to show you information on this figure. Anyone want to shout one out? Everywhere? Yes. Genes, yeah, what else? Interactions, yes. Food web structure, excellent, very good. So yes, we can find information, we can quantify it using information theory. People are studying it in the context of information at all these scales. So I should say information processing is the storage, the copying, and the use of information. It's a 4-step process: storage, translation, use, and copying. Not quite right. I'll get it in a second. But anyway, these 4 steps, they're abstracted steps. So how they actually occur, there's no specific machinery required to store information or to copy it or to use it. Those steps, though, happen in all kinds of different systems, from computers to protein synthesis. So within organisms, we have DNA and protein synthesis. Those are doing those 4 steps of information processing, and we use information theory to study that. Organisms and tissues within organisms even are sensing their environment. They're receiving chemical, visual, auditory cues. They're acting on those cues within their environment, so there's more use. They're storing information in their brains and bodies. Information processing happens, of course, in the reproduction and population growth processes. And again, we already use information theory to understand this. Network structure, as you mentioned, food webs. Biodiversity, we use information theory to describe the diversity of systems. So we have information processing across scales, but what we don't yet have, or even know, as far as I know, is how to compare and relate these different types of information in nature. So this turns out to be, as far as I can tell, a frontier, both in ecology and biology, even in physics. We don't have a way to convert the meaning that a daphnia takes from its environment into information and then somehow compare it to the genetic information in that daphnia. That, I think, may be possible someday, but the theory is not there yet. So if we want to view nature and ecology as this interaction of information processing and energy flows, we already know that we have the elements in our systems, and we're already studying them, but we lack the basic theory to know how to put these together. Except we can have, and here's what we can talk about later, we have many cases where we can be doing that, and I want to understand those cases better and look for commonalities among them to see if we can learn something. But for now, we'll go ahead. So my point there is a unified science has to incorporate energy, matter, and information dynamics. That will get us across scales. So my proposal for the unified science for living systems is, first of all, acknowledge that ecology has some kind of more dynamic definition, maybe growth, change, and persistence. And it's focusing on living systems. And this releases us from fixating on organisms and populations and communities as existing structures. We don't have to start with those anymore, and that's quite liberating. And to start bridging these gaps between metabolic and information processing theory. And I got here because the research that I will tell you about has all focused on the metabolism side of things. And that gets us, I think, quite farther than I thought before, but it will never get us all the way to what we want to know. And the reason is biodiversity, but that's not the last word. That's the first word, but we actually don't know very well how to get past that right now. Okay, shifting gears to the temperature work that I have done and been excited about for a long time. Basically building on this model of thinking about these fundamental, highly conserved metabolic processes, the way that they respond to temperature is very, they respond to temperature in the same way regardless of who's carrying them. So the photosynthetic machinery, the respiration machinery within cells accelerates as it gets warmer exponentially, and it doesn't matter whether that's happening in a plant or an animal or whatever else. So let's start there, because ecology is really complicated, and we've got zillions of experiments where we show that warming things changes things, but we really don't have a way to understand what that all means together. So let's start with what we know and see how far that gets us into the complexity of these systems. Right, starting with organelles, and we'll see how far we can get up through these levels of complexity. Okay, so I'm drawing on metabolic theory of ecology here, and some things that you may have seen before, and I'll just show you why I think we can start at this level and go up. This is Joey redrew this figure from Galuli's first paper on this, and this just shows that when you take out the effective body size on metabolic rate, and you put it on a log scale, you take a characteristic metabolic rate for a species given its size and its temperature. So gross, averaging out over all kinds of things. We see that as temperature increases, the metabolic rates at this sort of species level or characteristic species level increases exponentially. And the slopes are fairly consistent across these different groups. And this is just a subset of the taxonomic groups he had there. But from vertebrates, plants, fish, and unicells, we get this sort of around 0.62. It's a little bit different for fish in this paper. So we have evidence that this is, and he argued that this was consistent with the temperature dependence on respiration at that sort of subcellular level. So that's the argument there. And we see that species level. And this was an amazing observation because it really sort of allowed us to think, well, what if that's true? What if that is true, what does that mean for the way the systems work between an organelle and what we would get by comparing across species? And that was just what I needed in grad school as an principle, because I was facing that skepticism about warming up ecological systems in 2003 when I was designing projects. So this is from a paper that I did looking at the effect of temperature on the development of marine larvae. So larvae do their developing, and their marine organisms do a lot of their development from egg to sort of some sort of juvenile stage in the water column, and they're also floating around, and that for many species is their only chance to disperse. So it's an ecologically, demographically really important life stage. And it's been studied for a century by marine ecologists. So we pulled together 79 of these lab studies in which individuals from a population were collected at one place, brought into the lab, put at different temperatures, reared up, and then you measure how long it takes them to develop. So of course in cold temperatures it takes a long time, and as it warms up they develop faster. That turns out to happen every single time when food's not limiting and there aren't other stressors involved. But when we log transformed it and put it on the Arrhenius axes, it turns out that the effective temperature within these species is exactly what Galuli found for the effective temperature across species. It's exactly consistent with this 0.65 value that it might be related to the temperature dependence of respiration. So these are the same data. These are the model fits to those data, but it's just replotted in this way. So there is lots of variation among species. And the key here is knowing 10 degrees doesn't tell me how fast an organism is going to develop, right? Lots of variation at 10 degrees. You can develop fast or slow. But if I know how fast something develops at 10 degrees, then I know how fast it's going to develop at 20 degrees or 22. And that's true for all the species, from fish to invertebrates. We had 6 different phyla represented here. So temperature tells us how biological systems change. It doesn't actually tell us how they are now. We need to know more about that, but it tells us an awful lot about how they change. That's what I want to know. So here we are up at the ecosystem level. There's been lots of work. This is from a group in England by Gab Yvon de Rocher. In this Nature paper, he had 9 panels. I'm just showing you 3 because they all look the same. Effect of this is ecosystem respiration rate for a bunch of different data from the field that they could find for all these different ecosystem types. As temperature increases, you get an exponential increase in net ecosystem respiration rate. So here we're at like a pond level, a lake level, a river level. You still see the same effect. So it looks like across these levels of organization, we're seeing this coherent signal of temperature in a very simple way, without modeling species interactions, without knowing the trophic structure, without knowing the biodiversity of these systems. But I was in a community ecology lab, and I guess I still am a community ecologist, so we just launched right over all of this ecology that is really supposed to be really important. So that's what we wanted to know is it really important? What happens when we put these two things together? So we've done a fair amount of work that I won't go into on interactions and interaction strength, but one of the things we had to do this work and show how temperature affects species interactions, we had to make a critical assumption about what temperature does to populations. And not only did we make this assumption, anyone else modeling the effects of temperature on species interactions had to assume what temperature does to carrying capacity. So that is, how does temperature affect the abundance of a population and its growth rate when that population is growing alone? We've got to know that before we can go here, even though we went here first. So here, where this was work done by Joey. I have a picture of her on the next slide, and she's right there in the front row. So this is one of the most important experiments I think that we have done because it fills this critical gap in this theory logic chain. So to look at the effect of temperature on populations, we're just going to use a logistic growth model where we can use observations of abundance over time and estimate the intrinsic growth rate of the population and the carrying capacity. So the way that we had done this in these models was to simply assume that intrinsic growth rate scales with metabolic rate and that it increases with temperature, and that carrying capacity declines. It's basically the inverse. And the logic there is that as temperature increases, the per capita, per time demand of these organisms is going to go up, so a given environment can support fewer of them in a given amount of time. There's tons of evidence for the effect of temperature on intrinsic growth rate. It does increase exponentially with temperature, as you would expect, being closely related to the metabolic processes of the system. But there were no experimental tests of the effect of temperature on carrying capacity. We thought we could address this with a literature review, and we pulled together 40 studies that you would think would be able to do this. And it turns out when we got dug in there, they didn't assume the right things about resource supply and individual access to resources and the kinds of things you'd have to assume to test this model. So just to be clear about the hypotheses in this experiment, we hypothesized that increased temperature accelerates per capita metabolic rate, so the oxygen and carbon fluxes, and we have specific slopes that we expect based on what we understand to be the temperature dependencies of these processes. And because of that, we expect that the carrying capacity is going to decline inversely to what metabolic demand increases. Okay, so there's Joey and Jen Sunday. They did in our lab. They used Tetraselmis tetraheli, so a marine. It took some searching to find the right organism to meet the assumptions of this model. And the species, they move around, so they do get, we can hope that they get equal access to resources. They don't settle on the bottom or clump, which would be a problem here. So using some lab assays, we have this expectation of increasing photosynthesis rate, so oxygen flux with temperature, and indeed observed a slope that's entirely consistent with that. So this is just a check. This is what we thought should be happening. Our hypothesis depends on this, and it's happening. Photosynthesis rate goes up with temperature, and so did respiration with a steeper slope. All right, so on to abundance. So the predicted slope based on our hypothesis is that carrying capacity, so the density of the population, should go down with increasing temperature with a slope that's the opposite of what we now know we saw for oxygen flux. And after tracking these populations and doing all kinds of work to count these individuals and raise them and have them not die, this is what she found. So kind of good, but not fully satisfying, right? Density goes down, but not exactly as we expected. So carrying capacity, this is in terms of density. This is what matters for evolution, population growth rates, the number of individuals. This is our link to evolution and biodiversity. There's another way to measure carrying capacity, though. You could measure it in terms of the biomass of the system, right? So biomass worked. Biomass behaved. The population biomass declined with increasing temperature, just as we expected. So this was a clue. Sent Joey back to the flow cam to do some size estimates. And what she found here was that cell size declined with warming by about 2. So we didn't have a theoretical prediction for this, but we know temperature size rule, declines in body size with warming, is extremely common. Empirically, this is pretty much about the rate that size declines with temperature. There's several explanations for why. We don't know exactly why it happened here, but it happened. And that just meant we had more smaller individuals at warm temperatures than we would have expected. So Joey then wrote this out math-wise, and she, to the simple metabolic equation that we've been working with, she made body size be temperature dependent. And this was based on what we observed empirically. So in MTE so far, there's very little interaction between body size effects and temperature effects. You can just factor one out and focus on the other. But what we learned is at the population scale, you can't do that anymore. So now when she took that empirical change in temperature, put it into K, reran the model to predict density as a function of temperature, now we see exactly, she was able to predict exactly what she saw. So she remodeled it, and the prediction matched the data. So this is exciting for several reasons. As I mentioned, it gives us some empirical data for something that we and others had been assuming for years. It also shows that when we want to take MTE into populations, we can no longer keep body size and temperature separate. And that allows us to get into some of the community and population ecology that we're interested in. It also allows us, even if you're not interested in population ecology and temperature, how the environment supports abundance of individuals is pretty much central to anything we are interested in knowing about ecosystems, communities, and how organisms can be supported by their environment. So this link, even though it was using a logistic growth model, actually conceptually then can now allow us to think about temperature and community dynamics in lots of other ways. So in the interest of time, I'm going to jump ahead a bit. I'm going to skip ahead to, we have spent a lot of time building up from the population into the community dynamics. But I'm going to jump ahead and tell you a little bit about variability. Because I started off with expectations, right? So what we're doing there is starting at the very beginning and trying to build up. So that right there gave us a reason. I mentioned earlier those phytoplankton studies in a warming ocean. Now we have a simple model to relate a warming ocean to phytoplankton abundance. And it should be more complicated, of course, to use it in the field, but we could start with something like that. But the field is much more complicated, and we want to know what to expect of warming. And there's a lot of effort, of course, to understand warming, ecological and biogeographic patterns as they result from warming. This is another project that came out of that NCEES group, and this is the Velocity of Climate Change paper showing that, of course, most of the world is warming, but also to retain your thermal environment, you need to move, most species, and here's how far and fast you have to move. And it really suggests that the tropics are not only warming faster, but you have to be able to move faster. So we know that many species or ranges are shifting, and we're studying that using species range, what we understand about ranges, and I'll get to that if I have time, but we also know that many species are not moving. So understanding how temperature affects the performance for species matters because many species are not moving. So does warming more negatively affect low latitude populations and species? This is something that's come out of a lot of work using thermal performance curves to jump up and project climate change impacts. And we quickly get to places, and there were seminal papers by Deutsch and Tewksbury and other people doing this and then finding that tropical species are more vulnerable to warming because the idea is they live near their thermal optimum. This is one of those ideas that's made it into the IPCC and into IPBEST by people who aren't staying on top of the literature anymore, and it's basically a truth. But it's not a truth that makes a lot of sense to me. So the one question is, are tropical species more vulnerable to warming than temperate species? We can examine this using a thermal performance curve, and we did a working group that I led on temperature and species interactions. This is a paper that was led by Dave Vasseur and all. So vulnerability to warming depends on the intersection of the thermal regime and the thermal performance curve. Here's a thermal performance curve. We can consider three types of thermal environment that this organism possessing this thermal performance curve might live in, all with this average temperature. It could be an average temperature with Gaussian variation, an average temperature with wider Gaussian variation, so a very variable environment, more like a tropical environment, and this is more like a temperate environment. or there could be a skewed thermal distribution. So how an organism with this thermal performance curve experiences an average temperature depends a lot on actually how that environment varies. And I'm just going to go a little bit quick to get through this, but we project forward these thermal regimes, pass it through a thermal performance curve, and get to expectations for performance. So temperature above this temperature is going to mean increased performance, exponentially increased. Temperature below this temperature is going to mean less performance by this organism. But if you exceed the thermal optimum, now you get also decreased performance. And you can just see you get different shapes of a frequency distribution of performance depending on the variability of thermal regimes. So when we take into account variability, we see especially that these very variable environments lead to distributions of performance that are generally much lower than what you'd expect based on. So using the same data set that Deutsch et al. And those original papers used, we then went back, we got the temperature, the thermal histories from where those insect species were collected. We got their thermal performance curves, which were in the Deutsch et al. And other papers. And we looked at how, given the thermal variation of the environments these organisms were from, were they in fact worse off being at the tropics compared to the temperate zone? So when we do that, here's performance. It was basically population growth rate as a function of latitude here. So here are the tropical species growing really fast, and here are the temperate species. If we increase the mean temperature, everybody grows a bit faster, except for a couple. If we change the standard deviation, there's pretty much temperate species seem to do better. And if we change skew, we get another sort of distribution of responses. So this is just changing the things, but we're really interested in climate change. So then we took these same thermal performance and the thermal history, and we used the IPCC scenario for future warming and said, well, given how mean, variability, skew, et cetera, are expected to change for these species, are tropical species really most vulnerable? And what we find is no, not at all. In fact, the temperate species are much more vulnerable under these future scenarios because they were well adapted to given the variation in their environment. Their average temperatures were halfway down their thermal performance curves. That's just because it gets really hot a lot in those places, perhaps, if they are adapted. I'm getting ahead of myself there. But if that's true, what we would predict is that these temperate species are much more vulnerable, and the tropical species are going to stay pretty close to their current fitness. So I'll just sum up that part by saying that's one example of showing, we have other examples, showing how just mathematics of a thermal experience. Given the biology of a thermal performance curve and the idea that it is the result of evolution and adaptation and physical constraints like that Arrhenius model, so given that species have that, we can use math, basically nonlinear averaging and knowledge of the thermal environment to make some projections about these species' performances. And when we do that, one of the parts I skipped, we also get different conclusions about which how thermal performance curves relate to species ranges. This is other work that Joey's done that shows that we do, in fact, over-predict ranges in warm areas if we fail to consider variation in temperature. So we are really working to make better expectations for climate change and doing so in an information-efficient way, which does sometimes require us to leave aside some of the complexity of the system. Okay, so to wrap up my whole pitch for science, ecological science for a changing world, this is how I see things now. I see this metabolic worldview where temperature is a huge aspect, constraint on how living systems change. And by understanding this better, we can understand how, so temperature for metabolic systems is like gravity, and it's like evolution for information systems. It's always happening. Living systems always have to deal with it. We're pretty used to that idea for evolution over here. I think maybe we forgot that for some of our ecological systems on that side. So I hope that by doing that, we can continue to strengthen this. So if your work is in this area and it sometimes feels irrelevant, it's not. It's really, really relevant. And I encourage you to keep going and help trace its consequences up through this process. Because as I said before, the science to policy process is only as good as its weakest link. And there's a lot of work going on in here now, and we need to make sure there's still really great work happening on that side of things. So we get motivated to skip ahead in this process, I think, because we're afraid, because we're worried about global change, because we want to make a difference. We want to be better at forecasting and projecting, but we are scientists and we can do great work by doing science. And I find such comfort in this perspective by Marie Curie that said, nothing in life is to be feared, it is only to be understood. So now is the time to understand more so that we may fear less.